So, for about 4 decades now, the field of psychiatry has basically been searching for the average brain of a patient with schizophrenia. We've been, averaging together thousands upon thousands of neuroimaging scans, just desperately looking for this one universal structural signature of the illness. But what if that average brain literally does not exist?
Right. What if it's a complete statistical illusion? Because, well, what if our algorithms have just revealed that we have been, statistically speaking, mushing together two completely different structural diseases this entire time?
Which is a terrifying but also completely thrilling thought, isn't it? I mean, it fundamentally changes how we view decades of clinical trials.
Oh, absolutely. The statistical ghost we've been chasing has essentially masked the biological reality of the actual individual sitting right there in our clinics.
Yeah, exactly. So welcome to our deep dive into the molecular brain imaging of schizophrenia. To really understand how we finally uncover that reality, I want to start by transporting you, the listener, into a really vivid, highly conceptual setting. So just imagine you are stepping into the final, highly secure chamber of a world-class neuroimaging institute.
A rather intimidating room, I would imagine.
Very. And standing right in the center of this massive observatory is a character we are going to call Medlock Holmes.
Huh.
So he's this brilliant investigator, and arrayed all around him are the most sophisticated tools of modern neuroscience.
And it is a truly formidable arsenal he has there. I mean, over in one corner, you've got the MRI, which is detailing the physical architecture of the brain, the cortical thickness, the entire gray matter landscape.
Right, static structure.
Exactly. And alongside it, there is diffusion tensor imaging, or DTI, which maps the really complex directional white matter highways, so the physical connections between all those gray matter regions.
And then over on the other side, there is the PET scanner, which is uncovering the hidden neurochemistry, right? The sort of invisible transactions of neurotransmitters that we can't see on an MRI.
Spot on. Plus, you have magnetic resonance spectroscopy, which is measuring the raw metabolites, the actual fuel and exhaust of the cellular machinery. And finally, functional MRI, which is watching the hemodynamic responses of these neural networks actually coming alive in real time.
But the problem, historically, is that Medlock Holmes had to use every single one of these tools in complete isolation. So, like, an MRI might reveal that a patient's ventricular volume is enlarged, but
it cannot tell you if the neurochemical transmission within the adjacent striatum is actually malfunctioning.
Exactly. Each technique essentially solved only a tiny, isolated fraction of the mystery. But in our imaginary observatory today, this room suddenly transforms. The maps produced by all these disparate machines are projected right into the center of the room. They're rendered transparent, and slowly they begin to overlap.
And that is where the magic happens, because suddenly the gray matter loss on the structural MRI aligns perfectly with the disrupted white matter tracts on the diffusion scan.
Yes, and the abnormal dopamine release that you visualize on the PET scan coincides directly with the impaired salience networks that are, flickering away on the fMRI.
And the glutamate abnormalities that we measure with spectroscopy correspond perfectly with dysfunctional hippocampal circuits.
Right, so the fragmented evidence finally forms a single coherent picture. And that is exactly our mission for this deep dive today. We are going to explore how we are finally integrating all these modalities to understand schizophrenia not just as a disease of, say, a single rogue neurotransmitter, but as a profoundly complex systems disorder.
To really ground this discussion, though, we do need to establish a core conceptual framework right from the start. The anchor for our analysis today, and frankly for all of psychiatry, is this. Psychiatry is the study of how brain, mind, and experience interact, and crucially, how that precise process can go wrong.
Absolutely. And for you, the listener, whether you are a psychiatry trainee, maybe a registrar preparing for your final board exams, or just a clinician striving to integrate the latest neurobiology into your daily practice, you should really consider this analysis a direct companion to Chapter 12.9 of Kaplan and Sadek's comprehensive textbook of psychiatry.
We highly recommend you read alongside that chapter. It will really help you build your mental models, clarify the mechanisms we discuss, and ultimately strengthen your clinical reasoning when you're actually on the ward.
Yeah, that textbook is essentially the spine of our discussion today. But we must offer a really critical clinical caveat immediately, right? Because Medlock Holmes still faces a rather fundamental limitation. If a patient presents to you with first episode psychosis today, can we just put them in one of these scanners and definitively diagnose schizophrenia?
The answer is unequivocally no, we cannot.
which is just an ongoing frustration for so many clinicians. I mean,
Oh, it's maddening.
right. A fracture is perfectly visible on a radiograph. A tumor shows up clearly on an MRI, yet psychiatric diagnostics remain entirely stubbornly clinical. I assume this is basically due to the sheer scale of individual variability.
That is the primary barrier, yes. While the group differences, say, if you're comparing a cohort of 100 patients with schizophrenia to 100 healthy controls, while those group differences are robust and statistically significant, the overlap at the individual level is just vast.
Right. So one person's scan might look completely typical.
Exactly. And furthermore, the neuroimaging abnormalities we do see in schizophrenia, well, they share significant variance with bipolar affective disorder, severe major depressive disorder, autism spectrum conditions, and, this is the really tricky part, even healthy individuals who simply carry a high polygenic risk score.
Wow. So if the specificity just isn't there for a diagnostic test, it does sort of beg the question of utility for the registrar on the ground. Like, why immerse ourselves in the intense physics and chemistry of PTT scans for the next hour if it won't give us a diagnosis?
Because neuroimaging is the ultimate engine for uncovering pathophysiology. It is the tool that bridges the gap between a purely phenomenological diagnosis and a biological one. It's the framework driving the discovery of biomarkers, the identification of ultra-high-risk prodromal states, and most importantly for everyday clinical practice, it is the absolute backbone of modern psychopharmacological drug development.
Right.
You simply cannot understand how your medications actually work if you do not understand the imaging that validated them in the first place.
That makes perfect sense. So then let us begin right at the foundation stone of that psychopharmacology, which is the dopamine hypothesis.
Yeah.
So Arvid Carlson's Nobel Prize-winning work originated, roughly 50 years ago? And it came largely from the sort of serendipitous clinical observation of chlorpromazine.
Indeed it did. Clinicians essentially noted chlorpromazine's profound, almost miraculous efficacy in attenuating acute psychotic symptoms. Subsequent biochemical research then revealed its primary mechanism of action, which was the antagonism of postsynaptic D2-like dopamine receptors.
So the initial logic was basically a simple deduction.
Very simple. The logic was, if receptor blockade mitigates the psychosis, then hyperactivity of the subcortical dopamine system must be the primary driver of the pathology itself.
Right, the too much dopamine theory.
Yeah.
But testing that hypothesis in a living human brain, I mean, that required a massive, unprecedented technological leap. And that milestone occurred in 1983, which was the very first time a living human was scanned to actually visualize neuroreceptors.
Yes, and the ingenuity of that experiment cannot be overstated. The researchers utilized spiperone, which is a potent first-generation antipsychotic, and they labeled it with a radioactive positron-emitting isotope, specifically carbon-11. This created the very first radiotracer of its kind, 11C3N-methylspiperone, or NMSP for short.
To just give our listeners a real sense of the logistical difficulty here, carbon-11 has a physical half-life of just over 20 minutes, so you cannot simply order a vial of it from a lab across the country.
No, absolutely not.
You literally need a cyclotron physically located next to the scanner, synthesizing the tracer, binding it to the drug, and injecting it into the patient before the radioactivity just decays into nothing.
It was an extraordinary feat of physics and radiochemistry, really. So, as the carbon-11 decays inside the patient's brain, it emits positrons. Now, these positrons travel a microscopic distance, just a few millimeters, before colliding with an electron in the tissue.
Right, the annihilation event.
Exactly. They annihilate each other and emit two gamma rays in exactly opposite directions. The PET scanner is essentially a ring of detectors that captures these simultaneous gamma rays, which allows the computer to map the precise spatial location of where the tracer is binding to the D2 receptors.
And just like that, molecular imaging was born. From there, we obviously evolved from NMSP to tracers like 11-cracklepride and eventually to these super high-affinity tracers like 18-felipride. But to critically read this textbook literature, clinicians really must understand the mathematical parameters these scanners output. You read a paper and it constantly refers to binding potential, Bmax, and KD.
And understanding those parameters is absolutely non-negotiable for interpreting target engagement. The overarching metric you will see reported in these studies is binding potential, or BP. Basically, BP is a proxy measurement for the density of available receptors in the specific brain region.
Right. So let's break this down into actual pharmacokinetics for a second. BP is derived from two distinct variables, Bmax and KD. If we think about this in terms of like competitive inhibition and receptor kinetics, Bmax essentially represents the theoretical maximum number of binding sites, right? It is the total receptor density in that tissue.
Correct. Imagine it as the total number of parking spaces available in a car park. And then KD is the equilibrium dissociation constant, which essentially represents the affinity of the tracer for the receptor. Crucially, KD is the inverse of affinity.
So a lower KD means the tracer has a very high affinity.
Exactly. It binds very tightly and dissociates very slowly.
So binding potential is essentially Bmax divided by KD. But here is the really critical variable that makes PT scanning so dynamic and honestly so fascinating. Our injected radioactive tracer is in constant, fierce competition with the patient's own endogenous neurotransmitters.
This is the core principle of competitive displacement. Think back to the car park analogy. If a patient is actively secreting highly elevated levels of endogenous dopamine, that native dopamine is already parked in a significant proportion of the D2 receptor spaces.
Right.
When we inject the tracer, like 11-cracklopride, the tracer arrives but cannot find any available binding sites. So it just washes out of the tissue, and the scanner records a low binding potential. And this happens even if the absolute total number of receptors, the Bmax, is perfectly normal.
This mechanism of competitive displacement is exactly how researchers transition from merely, counting static receptors to measuring dynamic real-time fluctuations of dopamine in living patients, which perfectly brings us to the most sophisticated model of this system, Anthony Grace's tonic versus phasic dopamine hypothesis.
Grace's model is brilliant because it provides the neurobiological nuance that the original simplistic dopamine hypothesis completely lacked. He divided dopaminergic transmission into two very distinct spatial and temporal domains, tonic and phasic.
Okay, let's unpack this, because I like to imagine dopamine regulation as a building's climate control system.
That's a helpful way to look at it.
Yeah, so you have the constant background thermostat, which is your tonic dopamine, and then you have the emergency fire sprinkler system, which is the phasic dopamine. So tonic dopamine refers to this steady baseline concentration of dopamine just sort of diffusing through the extrasynaptic space, providing a constant background tone.
And this tonic background tone is actually regulated by prefrontal cortical inputs. Crucially, it acts upon presynaptic autoreceptors to provide a continuous regulatory brake on the dopamine neurons themselves. It essentially prevents them from firing indiscriminately.
Conversely, phasic dopamine refers to those rapid, high-amplitude bursts of dopamine released directly into the synaptic cleft. And this happens in response to a salient stimulus, right? Like a sudden reward, a physical threat, or a really cognitively demanding task.
Yes. So the question becomes, what is the actual dysregulation in schizophrenia according to Grace's hypothesis? Because the old model just assumed there was too much dopamine everywhere all the time.
Which didn't make sense clinically. So what did Grace propose?
Grace posited a paradoxical relationship. He hypothesized that in schizophrenia, there is actually a primary deficit, a significant decrease in the tonic extrasynaptic dopamine.
Wait, a decrease.
Yes. And because this background dopamine is abnormally low, the physiological on the presynaptic dopamine neurons is severely compromised. The neurons essentially become highly sensitized.
Ah, I see. The system perceives this false baseline of dopamine depletion, so it basically primes all the presynaptic terminals, getting them ready to fire. And then when a stimulus does arrive, the ensuing phasic burst, that intrasynaptic dopamine release, is just wildly exaggerated.
Precisely. A hypodopaminergic background state directly generates a hyperdopaminergic synaptic explosion.
Which, when you think about it, maps absolutely perfectly onto the clinical phenomenology. If every single minor environmental stimulus triggers a massive phasic dopamine burst, the patient experiences aberrant salience.
Exactly. Neutral stimuli, like a passing car or a television advert, suddenly become imbued with intense, threatening, or grandiose meaning. And that is exactly what drives the formation of delusions.
It's an elegant theoretical model, truly. But proving it in humans required inducing some intense pharmacological stress tests while the patients were inside the PET scanners. So let us examine how they proved the phasic hyperreactivity part first.
They utilized an amphetamine challenge, didn't they?
They did, and the methodology is brilliant in its simplicity. Amphetamine acts as a potent psychostimulant. It literally forces the presynaptic terminal to reverse its transporters and just dump its vesicular dopamine stores directly into the synapse. Okay, so researchers first scan the patient at baseline with 11-cracklopride to establish their resting binding potential. Then, while they are in the scanner, they administer a dose of amphetamine.
Correct. The amphetamine triggers a massive release of endogenous dopamine, which, as we discussed, competitively displaces the 11-cracklepride from the D2 receptors. The scanner then reads a sudden drop in the radioactive signal. And the magnitude of that drop is your measurable dopamine release, or DAR.
And the results?
When comparing cohorts, the patients with schizophrenia demonstrated a significantly larger drop in raclopride binding following the amphetamine administration compared to healthy controls. Their amphetamine-induced DAR was demonstrably massively elevated, which definitively confirmed the hyperreactive phasic component of Grace's model.
I do want to interject a really nuanced point from the textbook literature here, just for clinical context. This exaggerated DAR is definitely a marker of dopaminergic dysregulation, but it is not entirely pathognomonic for schizophrenia.
That's a very important caveat.
Yes, studies have shown that patients with Tourette syndrome, particularly those with comorbid obsessive-compulsive features, they also exhibit significantly elevated DR following an amphetamine challenge. So it really highlights that we are imaging a circuit-level vulnerability here, not a specific DSM diagnostic label.
A vital distinction for clinical reasoning, absolutely. However, within the context of schizophrenia research, this challenge definitively proved the phasic excess. But that leaves the other half of the equation. How do we prove the first half of Grace's hypothesis? How do you prove that the baseline tonic dopamine is actually depleted and that the total receptor density is altered?
Right, because you cannot just stimulate the system to find that out. You have to deplete it entirely. And this requires the AMPT challenge.
AMPT, or alpha-methylparatyrosine, is a reversible inhibitor of tyrosine hydroxylase, which is the absolute rate-limiting enzyme in catecholamine synthesis. So administering AMPT temporarily halts the brain's ability to manufacture new dopamine. It effectively drains the presynaptic stores and clears virtually all the endogenous dopamine from the synapses.
Wow. So by clearing away all that endogenous competition, every single D2 receptor in the striatum is suddenly unmasked and fully available for the radiotracer.
Exactly. So when researchers compared the pre-AMPT baseline scans to the post-AMPT depletion scans, the elevation in binding potential was markedly greater in patients with schizophrenia.
Meaning there was more endogenous dopamine hiding those receptors at baseline.
Precisely. This confirms that their baseline resting intrasynaptic dopamine was indeed dysregulated. But furthermore, with all the endogenous dopamine completely removed, they could measure the absolute Bmax, the total density of D2 receptors. And it was shown to be genuinely, albeit modestly, elevated in the patient cohort.
Which suggests a structural compensatory upregulation. The brain senses this chronic deficit in tonic dopamine, so it builds more postsynaptic D2 receptors in this sort of futile attempt to capture more signal. But ironically, that only exacerbates the impact of those exaggerated phasic bursts when they do happen.
It's a vicious cycle. And we also have presynaptic imaging confirming the upstream dysfunction as well. Researchers utilize 18-FF-opia, which is a radio-labeled analog of L-DOPI.
Now, this tracer is different, right? It does not bind to receptors at all.
No, it doesn't. It is taken up by the presynaptic terminal and metabolized by the enzyme dopa-decarboxylase.
So 18-FF-DOP is effectively measuring the manufacturing capacity of the dopamine neuron itself. It's looking at the factory, not the parking spaces.
That's a great analogy, and meta-analyses consistently demonstrate that 18-FFDO uptake is significantly elevated in the striatum of patients with schizophrenia. And fascinatingly, it's even elevated in their asymptomatic first-degree relatives.
So the presynaptic machinery is just an overdrive from the start.
Yes, synthesizing dopamine at an accelerated rate, essentially loading the vesicles, just waiting to fire those hyperreactive phasic bursts.
The dopamine data is incredibly comprehensive, isn't it? But here is where we really must challenge the historical paradigm. Because if dopamine hyperactivity is purely a downstream effect, if it's just the alarm bell ringing, what is the actual fire causing it? The dopamine hypothesis alone fails to adequately explain the profound negative symptoms and the cognitive decline we see in schizophrenia.
It does. It falls completely short there.
Which drives us into body section 2 of our deep dive, the multimodal chemical symphony, starting with glutamate.
The glutamate hypothesis shifts the primary site of pathology away from the subcortical striatum and up into the prefrontal cortex. Glutamate is, of course, the central excitatory neurotransmitter in the brain. And the prevailing model here suggests a hypofunction of NMDA receptors, specifically those located on parvalbumin-containing GABAergic interneurons up in the cortex.
Okay, let me just trace the circuitry here aloud to make sure I have this mental model right for our listeners. So normally, these GABAergic interneurons provide strict inhibitory control over the descending glutamatergic pathways, that project all the way down to the ventral tegmental area in the midbrain, right?
Yes, they act as the brakes.
So if the NMDA receptors on those specific interneurons are malfunctioning, the interneurons fail to fire.
Which means those descending excitatory pathways completely lose their inhibitory regulation. They become overactive, and they subsequently overstimulate the midbrain dopamine neurons. This directly drives the striatal hypergopaminergia. So the hypofrontality, the quiet cortex, directly causes the subcortical excess.
It's just incredible how it's all connected. And the clinical evidence for this stems largely from ketamine, does it not? Ketamine is an NMDA receptor antagonist, binding specifically to the PCP site within the ion channel. And decades of clinical observation have shown that subanesthetic doses of ketamine administered to healthy individuals produce this transient state that is virtually indistinguishable from acute schizophrenia.
And crucially, unlike amphetamine, which primarily only mimics the positive symptoms, like the paranoia ketamine induces the positive symptoms, plus the negative symptoms like emotional blunting and the profound cognitive disorganization. It produces the full syndromal picture.
Which is wild. And to prove this mechanism in vivo, researchers combined the ketamine challenge with p-imaging. And this is where I find the methodology just absolutely fascinating. They administered a glutamate antagonist, right? But they scanned the patients using 11-cracklepride, which is a dopamine tracer.
Yes, they did.
Wait, if ketamine acts entirely on the glutamatergic NMDA receptor, why would a dopamine tracer show any displacement at all? Those are two completely distinct neurochemical systems.
But that is precisely the point they were trying to prove, the direct causal link between those two systems. When healthy volunteers were administered ketamine, effectively blocking their cortical NMDA receptors, their 11-cracklepride binding down in the striatum dropped precipitously up to 17% in the ventral striatum.
Because blocking the glutamate receptor functionally released the brake on the dopamine neurons, causing a massive spike in endogenous dopamine that then kicked the raclopride off the D2 receptors.
Precisely. It is direct, visualized proof of the circuit in a living human. Furthermore, the magnitude of that raclopride displacement correlated strongly with the severity of the transient psychotic symptoms, the perceptual alterations, and the thought disorder that the volunteers actually experienced while inside the scanner.
It fundamentally reshapes how we view the illness. It's not just a dopamine problem, it's a network problem. But the symphony involves more than just dopamine and glutamate. Let us pivot to serotonin. The serotonin hypothesis originated from a completely different vector, didn't it? Not from an antipsychotic medication, but from a potent hallucinogen.
Yes. The structural similarity between native serotonin and lysergic acid diethylamide, or LSD, sparked the early hypotheses here. If a serotonergic agonist induces profound visual hallucinations and altered reality testing, perhaps endogenous serotonin is heavily implicated in the pathophysiology of idiopathic psychosis.
And this theory gained monumental clinical traction with the advent of second-generation atypical antipsychotics. Because while haloperidol primarily targets D2 receptors, drugs like clozapine, risperidone, and olanzapine are largely defined by their potent antagonism of the 5-HT2A receptor.
To investigate this, the imaging field had to develop highly specific radiotracers for the 5-HT2A receptor, such as 18-foltanserin. Now, the results in drug-naive or completely unmedicated patients with schizophrenia have been, well, highly heterogeneous.
Like what way?
Some studies indicate significant decreases in 5-HT2A receptor density in the prefrontal cortex, whilst others report absolutely no significant deviation from healthy controls at all.
What about the other receptor subtypes? Because serotonin is a notoriously complex system. There are at least 14 distinct receptor subtypes.
True. Researchers have also heavily focused on the 5-HT1A receptor, utilizing tracers like 11CWA100635, which is a potent antagonist. And the findings here are equally complex. We see region-specific alterations. For instance, increased binding in the medial temporal cortex alongside decreased binding in the amygdala.
It's all over the place.
It is. While the primary lesion may not inherently be serotonergic, molecular imaging confirms that the serotonergic architecture is undoubtedly distorted in these patients, which elucidates exactly why dual-acting dopamine-serotonin antagonists provide such robust, broad-spectrum clinical efficacy.
Right, covering more bases. Now, let us examine a clinical observation that literally every registrar encounters immediately upon entering a psychiatric ward: the staggering prevalence of heavy tobacco smoking among patients with schizophrenia.
It's ubiquitous.
It is, and it often appears driven by this profound compulsion suggestive of intense self-medication rather than mere habituation or boredom.
This epidemiological observation actually directed investigators towards the cholinergic system, specifically the nicotinic acetylcholine receptors, or NHARs. Postmortem analyses had long indicated marked deficits in the alpha-7 nicotinic receptor subtype in these patients.
Why is the alpha-7 subtype so functionally critical, though? What does it actually do?
The alpha-7 receptor is heavily concentrated in the hippocampus and the prefrontal cortex, and it is a fundamental mediator of something called sensory gating.
Ah, sensory gating.
Yes, sensory gating is essentially the brain's neurophysiological mechanism for filtering out repetitive, irrelevant environmental noise. Think of the cocktail party effect, your ability to focus on one conversation while ignoring the background chatter. In schizophrenia, this gating mechanism is severely impaired. The patient is chronically inundated by all sensory stimuli simultaneously.
Which obviously contributes massively to cognitive fragmentation and sensory overload. It must be exhausting.
Indeed. And using novel PIT tracers like 18-FACEM, researchers have finally quantified this in living patients.
What do they find?
The PEAT data confirms the postmortem findings beautifully. Patients with schizophrenia demonstrate a significantly reduced volume of distribution, which is a metric quite analogous to binding potential of these alpha-7 receptors. This provides a robust biological rationale for the heavy smoking behavior.
So the intense nicotine intake is quite literally a physiological attempt to flood these deficient alpha-7 receptors, temporarily restoring a degree of sensory gating and allowing them some cognitive focus.
Precisely, which highlights a distinct pathway for novel therapeutics. If we can isolate the cognitive deficit to the alpha-7 receptor, we can develop targeted agonists to address the cognitive symptoms, a domain where our standard D2 antipsychotics fail miserably.
That's incredible. And moving to another highly relevant clinical domain, particularly regarding adolescent risk factors, we have to discuss the endocannabinoid system. The epidemiological link between early heavy cannabis use and the subsequent development of psychosis is very well established at this point.
It is.
So what does the PET data actually reveal regarding the cannabinoid receptor type 1, the CB1 receptor, in patients who already have established schizophrenia?
Well, the CB1 research is honestly a masterclass in the methodological challenges of neuroimaging. Researchers have utilized several tracers here, notably 11 COMAR and 18 FFM-PEP2. Now, initial pilot studies suggested massive elevations in CB1 receptor binding, particularly in the ventral striatum, which seemed to correlate strongly with negative symptom severity.
Okay, elevated receptors. But wait, didn't subsequent much larger cohort studies report the exact opposite? Like widespread decreases in CB1 binding across the amygdala, caudate, and hippocampus? How do we reconcile such wildly contradictory data? Are the tracers just inherently unreliable?
No, the tracers themselves are kinetically very sound. The discrepancy actually stems from the immense biological heterogeneity of the sample populations they used, specifically regarding sex differences.
Sex differences.
Yes. Methodological rigor eventually revealed that in healthy populations, males exhibit approximately 41% higher CB1 receptor availability than females at baseline.
Wow, a 41% baseline variance is enormous. So if a study inadvertently skews its sex ratio, say, testing mostly men in the patient group and mostly women in the control group, without rigorous statistical correction, it will just completely obliterate the true disease-specific signal.
Precisely. It's a massive confounder. Once researchers utilized third-generation tracers and strictly statistically accounted for sex and prior cannabis exposure, the consensus shifted dramatically. The current reliable data points toward a net reduction in CB1 receptor levels in schizophrenia.
Interesting.
Furthermore, in male patients specifically, greater reductions in CB1 availability are closely associated with more severe psychopathology and significantly poorer cognitive performance.
That is such a crucial lesson in experimental design. Now, before we transition to the direct clinical application of these findings on the ward, we need to address a major contemporary debate in the literature: neuroinflammation.
Oh, yes.
The hypothesis that schizophrenia, or at least the acute psychotic exacerbation, is basically driven by microglial activation and a localized cytokine storm in the brain.
The neuroinflammatory hypothesis is highly compelling, there's no doubt, particularly for early-stage or treatment-resistant illness. And the field has heavily relied upon TSPO-PE imaging to investigate this.
TSPO?
Yes, TSPO is an 18-kiloday translocator protein located on the outer mitochondrial membrane. Crucially, its expression is dramatically upregulated when microglia, which are the brain's resident macrophages, are activated by inflammation.
So logically, a high TSPO signal on a P scan should theoretically equate to active neuroinflammation in that brain region. But the literature seems really fractured on this point as well.
The TSPO literature in schizophrenia is fraught with methodological peril, frankly. Firstly, the signal-to-noise ratio for many of these TSPO tracers is inherently variable, but the critical fatal flaw for many of the early studies was actually a genetic one.
A genetic flaw.
Yes. Humans carry a specific single nucleotide polymorphism, the RS6971 polymorphism, which completely dictates the binding affinity of the TSPO protein itself.
Oh, I see. So the tracer binds differently depending on the patient's genetics, completely, regardless of whether they actually have active inflammation.
Exactly. This polymorphism creates high affinity binders, low affinity binders, and mixed affinity binders in the general population. If you scan a cohort without running a priori genotyping on every single person, your data is entirely confounded by this genetic variance. You must genotype every participant and typically exclude the low affinity binders entirely just to achieve valid, comparable quantification.
So when modern studies actually do that, when they rigorously control for the RS6971 polymorphism, what does the meta-analytic data actually show regarding TSPO in chronic schizophrenia?
The evidence for elevated microglial activation in chronic schizophrenia via TSPO is exceptionally weak. In fact, some rigorously controlled recent reviews suggest a potential decrease in TSPO binding. The general consensus is rapidly moving away from TSPO as a reliable, universal biomarker for the disorder.
Does this invalidate the neuroinflammatory hypothesis entirely, though?
Not at all. It merely invalidates our previous method of measuring it. The field is now pivoting to much more specific targets, such as 11CS1P1, which targets the sphinxine 1 phosphate receptor 1.
Okay, what does that do?
It's a key regulator of immune cell trafficking and neuroinflammation. Groundbreaking work by Chan and colleagues has shown S1P1 elevations in postmortem schizophrenia tissue, but crucially, these elevations appear restricted to a very specific structural subtype of the illness, which is a concept we will explore deeply when we discuss artificial intelligence in a moment.
Right, the AI subtyping, which is mind-blowing. But first, let us ground all this molecular chemistry firmly onto the clinical ward. This brings us to body section 3. The integration of clinical reasoning and the therapeutics of occupancy.
This is where it gets highly practical.
Very. Because for this psychiatric registrar tasked with titrating an antipsychotic for an acutely psychotic patient tonight, what does all this PT imaging actually tell us about how to dose? We know we need to block D2 receptors, but exactly what percentage is required for actual clinical efficacy?
Through decades of meticulous target engagement studies, the field has precisely defined the therapeutic window. For first-generation antipsychotics and the vast majority of atypical agents, reliable clinical efficacy, specifically the attenuation of positive psychotic symptoms, requires occupying between 60% and 80% of the striatal D2 receptors.
Okay, so 60 to 80%. If the occupancy falls below 60%, the dopamine, blockade is basically insufficient to halt those exaggerated phasic bursts we talked about, and the psychosis simply persists. But what happens when we breach the 80% threshold? Is more better?
Absolutely not. Pushing striatal D2 occupancy above 80% does not yield any additional antipsychotic benefit whatsoever. It merely saturates the receptors and induces severe toxicity.
Toxicity in what form?
Specifically, exceeding 80% occupancy virtually guarantees the emergence of extrapyramidal side effects, or EPS. We're talking about Parkinsonian rigidity, acute dystonias, and the profound, agonizing restlessness of akathisia. It also drastically increases the risk of hyperprolactinemia due to simultaneous D2 blockade in the tuberoinfundibular pathway.
Let's apply this mathematical rule to a really ubiquitous clinical scenario, something every doctor sees, prescribing oral risperidone. How does the plasma concentration of risperidone correlate with this PT occupancy data?
Well, when a clinician prescribes a standard evidence-based oral dose of 3 to 4 milligrams of risperidone daily, the steady-state plasma concentration typically ranges between 15 and 40 nanograms per milliliter.
And what does the PET scanner reveal at that specific plasma concentration?
At 15 to 40 nanograms per milliliter, the striatal D2 occupancy rests perfectly within that 70% to 80% therapeutic sweet spot. The patient receives maximal antipsychotic efficacy with a very minimal burden of motor side effects.
However, clinical desperation often leads to dose escalation, right?
Yeah.
A registrar might see a patient still experiencing auditory hallucinations on 4 milligrams and decide, well, I'll push the dose to 6 or 8 milligrams daily, just hoping for a more robust response.
And the PET data explicitly demonstrates the biological futility of that decision. At 6 milligrams daily, the plasma concentration reliably exceeds 40 nanograms per milliliter. The PE scan confirms that at this concentration, the striatal D2 occupancy crosses that 80% toxicity threshold. You are no longer treating the psychosis, you are simply inducing iatrogenic Parkinsonism.
Which is such a vital lesson. And the exact same pharmacodynamic principles apply to long-acting injectable or LAI formulations. When guidelines recommend a 25 to 50 milligram fortnightly dose of LAI risperidone, it is precisely because that specific dose range maintains plasma levels within that 15 to 40 nanograms per milliliter window, sustaining optimal receptor occupancy without breaching the EPS threshold.
Exactly. When early trials tested much higher LAI doses, the plasma levels spiked, the EPS incidence absolutely soared, and those doses were rightfully abandoned. PET imaging has fundamentally transformed psychiatric prescribing from empirical, blind trial and error into actual precision pharmacology.
Yet there's one glaring, monumental exception to this 60 to 80% rule. Clozapine.
Clozapine.
Clozapine is universally recognized as the gold standard for treatment-resistant schizophrenia. It is arguably our most efficacious antipsychotic, full stop. Yet, when early PP researchers scanned patients on clozapine, the data was entirely contradictory, wasn't it?
It was baffling. This was termed the clozapine paradox. Early occupancy studies utilized 11-craclopride, as we discussed earlier. When they scanned patients who were stabilized on highly effective doses of clozapine, the raclopride binding indicated that clozapine was only occupying roughly 40% to 50% of the striatal D2 receptors.
But according to the established rule we just outlined, 40% occupancy should be clinically useless. Furthermore, clozapine induces virtually no extrapyramidal side effects. So how on earth do we reconcile its profound clinical efficacy with its apparent failure to engage the target?
The paradox, it turned out, was an artifact of the radiotracer's limitations. 11-craclopride is a moderate affinity tracer. It is highly effective for quantifying receptors in the striatum, where D2 density is immense. However, it completely lacks the signal-to-noise capability to accurately map D2 receptors out in the cortex, where they are very sparsely distributed.
Right, so they needed a much more sensitive instrument to visualize the extrastriatal receptors.
Exactly. The resolution of the paradox finally arrived with the development of high-affinity radiotracers like 18-falipride. When researchers rescanned those exact same clozapine patients using 18-falipride, the true pharmacological profile emerged.
And what was it?
The low occupancy in the striatum was confirmed, which perfectly explains the complete absence of EPS, as clozapine largely spares the motor circuitry. But when they looked outside the striatum,
Out into the temporal and limbic cortex.
the extrastratocortical D2 occupancy was significantly higher. Clozapine demonstrates preferential regional binding to cortical D2 receptors. This robust extrastatical target engagement is now posited as the main mechanism underlying its superior efficacy, particularly in ameliorating cognitive disorganization and negative symptoms, while its weak striatal binding spares the patient from motor toxicity.
Clozapine shares a qualitatively similar, albeit less potent, regional binding profile, right?
Yes, it does.
This spatial conceptualization, imagining risperidone functioning as a sort of blunt instrument blanketing the striatum versus clozapine acting as a highly targeted intervention for the cortex, is just a vital mental model for any clinician selecting an antipsychotic. And this PE technology is now the mandatory gateway for all novel drug development.
Oh, absolutely. No psychotropic agent advances through phase 1 or phase 2 clinical trials today without rigorous PE target engagement validation. Take aripiprazole, for instance, a D2 partial agonist. PE was instrumental in proving that it occupies the receptor site sufficiently to block excessive phasic dopamine bursts, whilst its intrinsic agonism maintains just enough basal signaling to prevent the severe hypodopaminergia associated with full antagonists.
The textbook outline also highlights blunanserin in this context.
Yes, blunanserin represents a fascinating refinement in receptor targeting. It possesses a high affinity not only for D2 receptors, but specifically for D3 receptors. Researchers utilized a very novel agonist tracer, 11C plus PHNO, to directly quantify its binding in living subjects, confirming robust D3 occupancy.
And why is D3 important?
Given the D3 receptor's dense localization in limbic pathways and its heavy implication in cognitive processing, proving this target engagement provides a very strong biological rationale for its unique clinical profile.
We are also witnessing PT validate entirely novel stereochemical approaches, right? Like the non-racemic enantiomers SCP-4199 and LB-102, PP ensures these highly precise molecular variants hit their intended targets without off-target toxicity.
It really represents the pinnacle of precision neurochemistry. But if we want to truly understand the future of schizophrenia research, we must move beyond the synapse itself and integrate the genetic code and the raw computational power of artificial intelligence.
Which brings us to body section 4. Genetics, AI, and machine learning subtyping. Let us delve into imaging genetics first. The heritability of schizophrenia is estimated at approximately 80%, which is massive, yet it is a highly polygenic disorder involving thousands of small effect variants.
Yes, it's not a single gene disorder like Huntington's.
Exactly. Molecular imaging allows us to study what are called intermediate phenotypes, visualizing how these specific genetic risk variants alter macroscopic brain function long before the onset of overt clinical disease.
Instead of a simplistic, frankly outdated model where gene X causes schizophrenia, imaging genetics allows us to make much more precise statements, like gene X induces a specific metabolic inefficiency within this specific neural circuit.
And a prime example of this from the literature is the COMT gene, which encodes catechol-methyltransferase. Now, this is the primary enzyme responsible for degrading dopamine in the prefrontal cortex, where dopamine transporters are actually quite scarce.
Researchers focus heavily on a specific functional polymorphism within this gene, the Val158 MET variant. Individuals who are homozygous for the Val allele possess a highly active version of the COMT enzyme.
Meaning it chews up dopamine much faster.
Exactly. It catabolizes prefrontal dopamine at a significantly accelerated rate, which results in chronically lower baseline cortical dopamine levels.
Which directly links back to Anthony Grace's model of a hypodopaminergic cortical state. But how does this specific genetic variant manifest functionally, say, on an fMRI scan?
Well, the prefrontal cortex relies on extremely precise dopamine signaling to maintain optimal signal-to-noise ratios during cognitive tasks, such as working memory paradigms like the N-back test, where you have to remember a sequence of letters or numbers.
Right.
Because the Val carriers have depleted prefrontal dopamine, their cortical signal-to-noise ratio is severely degraded. Consequently, when asked to perform a working memory task, their fMRI scans reveal a profound inefficiency.
What does that look like on the scan?
The brain literally has to work harder. It must recruit vastly wider auxiliary neural networks and expend significantly more metabolic energy just to achieve the exact same behavioral performance as someone with the MET variant.
And crucially, these genetic variants do not operate in a vacuum. They interact in complex, nonlinear ways, a phenomenon known as epistasis.
A landmark study really beautifully illustrated this by examining the epistatic interaction between that COMT Val158 MET variant and a completely different polymorphism in the GRM3 gene, which modulates glutamatergic synaptic transmission.
What did they find?
They discovered that carrying the high-risk GRM3 variant induced severe prefrontal inefficiency, but only if the individual also possessed the Val homozygous' COMT background.
So if the individual carried the MET allele instead, it actually buffered the glutamatergic risk.
Yes, the genetic vulnerabilities compound each other, ultimately destabilizing the entire circuit.
We can even detect this biological vulnerability in asymptomatic relatives, can't we? When researchers administer 18-F-etopa to first-degree relatives of patients with schizophrenia, their striatal uptake is actually elevated compared to healthy controls.
Yes, they inherit the hyperactive presynaptic dopamine manufacturing capacity, essentially the loaded weapon, even if environmental factors never pull the trigger to induce full psychosis.
Which finally brings us to the most formidable obstacle in psychiatric research, and honestly the conceptual breakthrough that frames our entire conclusion, the problem of heterogeneity. For decades, every clinical trial and imaging study has operated on the assumption that schizophrenia is a single, albeit highly variable, neurobiological entity.
And that assumption might be our greatest failure.
I use an analogy to highlight the absurdity of this approach. Imagine researchers attempting to determine the average physiological phenotype of a human being by placing 20 individuals in a room. However, unbeknownst to the researchers, 10 of those individuals are toddlers, and 10 are elite professional basketball players.
Right. If you apply standard statistical averaging to that heterogeneous group, you will conclude that the average human in that room is roughly 5'5" tall, with moderate cardiovascular output.
But a 5'5" human literally does not exist in that room. By averaging two fundamentally distinct populations together, the researchers have created a complete statistical phantom. They have mathematically erased the biological reality of both the toddlers and the athletes.
And this is precisely the methodological error that has plagued schizophrenia neuroimaging for 40 years. We take highly diverse patient cohorts, we average their structural MRI scans, compare them to healthy controls, and we dutifully report modest, widespread cortical thinning.
But that phantom average provides almost zero predictive value for the actual individual patient sitting in front of you in the clinic.
Exactly. Enter machine learning and the paradigm-shifting work of Ganesh Chand and various international imaging consortiums.
So how did their application of AI actually circumvent this statistical trap?
They completely abandoned the traditional case control averaging model. Instead, they utilized a technique conceptually similar to normative modeling. First, the machine learning algorithm was fed thousands upon thousands of scans to learn the precise multidimensional parameters of a structurally healthy brain across all ages and demographics.
So it established a rigorous AI-driven baseline of normality first. And then how did it analyze the patient cohort?
Instead of comparing the schizophrenia patients to each other or averaging them together, the algorithm evaluated every individual patient's scan against the normative model. It asked one question. In what specific multidimensional vector does this individual brain deviate from the healthy baseline? It mapped the unique structural deformations of every single patient one by one.
It mapped the individual deviations, unblinded the data, and what did the algorithm actually reveal? Did it find the statistical phantom, or did it finally separate the toddlers from the athletes?
It decisively shattered the concept of a single disorder. The AI identified two fundamentally distinct, totally non-overlapping neuroanatomical subtypes of schizophrenia that had been hidden within our statistical averages for decades.
Okay, let us dissect these subtypes because this is revolutionary. What defines subtype 1?
Subtype 1 constitutes the majority, encompassing approximately 63% of the patient cohort. Structurally, this subtype is defined by widespread, severe reductions in both gray matter volume and white matter integrity across the entire cerebrum.
And clinically?
Clinically, this profound structural degradation correlates tightly with a longer duration of illness and significantly poorer pre-morbid educational and social functioning. It aligns perfectly with the classic neurodevelopmental or neuroprogressive tissue loss model of the disease.
So roughly 2/3 of patients exhibit pervasive cortical atrophy. What about the remaining 37%? What is the architecture of subtype 2?
Subtype 2 is the absolute paradigm breaker. In this substantial minority, nearly 40% of the sample, the algorithm revealed that their overall widespread cortical gray and white matter volumes were entirely indistinguishable from healthy controls. They completely lacked the pervasive atrophy seen in subtype 1.
Wait, their global brain volume is normal, but they carry a diagnosis of severe schizophrenia.
Their global volume is normal, with one highly specific, glaring exception. While the cortex remained intact, these subtype 2 patients exhibited enlarged striatal and internal capsule volumes. Their basal ganglia structures were significantly hypertrophied compared to healthy controls.
Consider the devastating implications of this for historical research. 63% of patients have smaller atrophied brains. 37% have overall normal brains with enlarged subcortical structures. If you run a standard classical imaging study and just average those two groups together,
the severe atrophy of the 63% completely statistically masks the subcortical hypertrophy of the 37%. The final published average simply looks like a milder, diluted version of subtype 1. Subtype 2, which is a distinct biological disease process, gets entirely mathematically erased from the literature.
this AI-driven subtyping is truly the dawn of precision psychiatry. I mean, if a first episode patient presents to the ward tomorrow, and we can run a structural MRI through this algorithm to categorize them immediately, it completely alters the treatment trajectory.
Oh, fundamentally. Perhaps subtype 1 requires aggressive neuroprotective or glutamatergic agents to halt the widespread atrophy, while subtype 2 requires highly targeted, potent D2 blockade for their specific subcortical pathology.
It also clarifies the previously contradictory data we discussed. Remember the neuroinflammation target, 11CS1T1?
Yes. Chan's subsequent integration of molecular and structural data suggests that specific inflammatory markers may map exclusively onto one of these AI-derived subtypes, rather than representing a universal feature of the clinical syndrome.
By fusing the molecular chemistry of Penny with the structural resolution of MRI and processing it through the unbiased lens of artificial intelligence, we are finally disassembling the monolithic label of schizophrenia and uncovering the distinct biological realities beneath.
It is the ultimate vindication of Medlock Holmes's methodology. No single clue solves the mystery. The truth only ever emerges in the overlap.
That is an immense volume of complex neurobiology to synthesize. Let us consolidate our journey and distill the key clinical takeaways. We began by unpackaging the physics of PEDI using Bmax and competitive displacement to validate Anthony Grace's model. A pathological deficit in tonic background dopamine that removes the physiological brake, unleashing exaggerated hyperreactive phasic dopamine bursts directly into the striatum.
We then explored the multimodal symphony beyond dopamine. We examined how ketamine-induced NMDA hypofunction in the cortex directly triggers that downstream dopamine flood. We discussed the serotonergic alterations targeted by atypical antipsychotics, the cholinergic alpha-7 deficits driving compulsive smoking as a form of sensory-gating self-medication, and the profound impact of sex differences on cannabinoid CB1 receptor quantification.
We anchored this data to the clinical ward through the therapeutics of occupancy. We defined the non-negotiable 60 to 80% striatal D2 occupancy window for standard antipsychotics, demonstrating how exceeding that threshold solely guarantees EPS. We then resolved the clozapine paradox, revealing its preferential extrastriatal temporal limbic binding profile via high-affinity tracers like 18-falipride.
Finally, we integrated genetics and AI. We saw how the COMT Val158 MET variant induces prefrontal inefficiency during working memory, and how machine learning has structurally bifurcated schizophrenia into at least two distinct diseases. Subtype 1, defined by widespread cortical volume loss, and subtype 2, defined by normal global volumes but enlarged striatal structures.
Right. Now, for the trainees and registrars preparing for board examinations, it is time for some active learning reinforcement. My esteemed colleague will now administer 6 single best answer questions drawn directly from the core mechanisms we have discussed today. I will read the stem, we will pause briefly to allow you to formulate your clinical reasoning, and then the expert will provide the correct answer and the rationale.
Let us begin. Question 1. According to Anthony Grace's conceptual model of schizophrenia pathophysiology, what is the primary neurobiological state of extrasynaptic or tonic dopamine and intrasynaptic or phasic dopamine when compared to healthy controls?
Take a moment to think about the thermostat and the fire alarm.
The correct answer is decreased tonic dopamine and elevated phasic dopamine. The pathological reduction in background tonic dopamine fails to stimulate regulatory autoreceptors, removing the inhibitory brake and leading to hyperreactive, exaggerated phasic burst upon environmental stimulation.
Spot on. Question 2. Which specific PDT radio tracer is most appropriate for visualizing extrastriatal D2 and D3 receptors, a technological advancement that was absolutely crucial for explaining clozapine's unique mechanism of action?
The answer is 18-falipride. While 11-creclopride is sufficient for mapping the densely packed striatum, the significantly higher affinity of 18-falipride is required to achieve an adequate signal-to-noise ratio in the sparse temporal and limbic cortex, revealing clozapine's preferential extrastriatal binding.
Excellent. Question 3. Based on decades of P-target engagement studies, at what specific percentage of striatal D2 receptor occupancy do extrapyramidal side effects, EPS, typically become highly probable?
The answer is above 80%. The therapeutic window for efficacy is 60 to 80%. Pushing the dose higher, for instance exceeding 6 milligrams daily of oral risperidone, saturates the receptors beyond 80%, yielding no further antipsychotic benefit while actively inducing motor toxicity.
Question 4. An AMPT alpha-methylparatyrosine challenge administered prior to a PET scan achieves which specific physiological effect to aid in quantifying baseline receptor density?
The answer is it pharmacologically depletes presynaptic dopamine stores by inhibiting tyrosine hydroxylase. This eliminates endogenous dopamine competition, unmasking the D2 receptors and allowing researchers to measure the true absolute baseline Bmax, thereby demonstrating the compensatory receptor elevation in patients with schizophrenia.
Brilliant. Question 5. According to Chand and colleagues' machine learning analysis of global imaging consortiums, what precise structural brain features characterize the minority, approximately 37%, subtype 2 of schizophrenia?
The answer is overall normal global cortical brain structure, accompanied by significantly enlarged striatal and internal capsule volumes. This sharply contrasts with the majority subtype 1, which exhibits pervasive, widespread gray and white matter reductions.
And finally, question 6. The administration of subanesthetic ketamine in human models causes a measurable decrease in 11-creclopride binding in the striatum. What specific neurochemical mechanism does this radiotracer displacement indicate?
The answer is an acute increase in intrasynaptic dopamine release triggered by NMDA receptor antagonism. Blocking the glutamatergic NMDA receptors removes descending inhibition, causing endogenous dopamine to flood the striatal synapse, which competitively displaces the radioactive raclopride tracer from the D2 receptors.
If any of those mechanisms felt unclear, or if you struggled with the reasoning, we strongly advise you to return to Chapter 12.9 of Kaplan and Sadok's comprehensive textbook of psychiatry to review the foundational literature.
Before we conclude, though, we must leave you with a final consideration regarding the implications of the artificial intelligence data we discussed.
Indeed, we have just established that machine learning has definitively proven that the clinical label of schizophrenia encompasses at least two fundamentally distinct structural diseases, which our statistical models have been blindly averaging together into a phantom entity for 4 decades.
It forces a profound reevaluation of the entire history of psychiatric psychopharmacology.
It really does. I want you to carry this thought onto the ward with you tomorrow. If there are two structurally and perhaps chemically entirely different diseases hiding under the single diagnostic umbrella of schizophrenia, how many of our failed clinical trials over the last 40 years didn't actually fail? How many abandoned, ostensibly ineffective experimental drugs were actually brilliant, miraculous, targeted therapeutics? But they appeared to fail simply because we gave the right medication to the wrong biological subtype.
A very sobering historical reflection, but ultimately it is the foundation for immense hope regarding the future of precision psychiatric medicine.
That is all for this deep dive. Keep critically observing the data, keep questioning the historical averages, and keep exploring the maps.